Chapter 2, Biological Classification. Since the dawn of civilization, there have been many attempts to classify living organisms. It was done intensively, not using criteria that were scientific but born out of a need to use organisms for our own use, for food, shelter, and clothing. Aristotle was the earliest to attempt a more scientific basis for classification. He used simple morphological characters to classify plants into trees, shrubs, and herbs. He also divided animals into two groups, those which had red blood and those that did not. In Linnaeus' time, a two-kingdom system of classification with Plantae and Animalia kingdom was developed that included all plants and animals respectively. This system did not distinguish between the eukaryotes and prokaryotes, unicellular and multicellular organisms, and photosynthetic green algae and non-photosynthetic fungi organisms. Classification of organism into plant and animal was easily done and was easy to understand, but a large number of organisms did not fall into either category. Hence, the two kingdom classification used for a long time was found inadequate. Besides gross morphology, a need was also felt for including other characteristics like cell structure, nature of wall, mode of nutrition, habitat, methods of reproduction, evolutionary relationship, etc. Classification systems for the living organism have hence undergone several changes over the time. Two plants and animal kingdom have been a constant under all different systems. The understanding of what groups organisms be included under these kingdoms have been changing. The number and nature of other kingdoms have also been understood differently by different scientists over the time. Cell type in Monera is prokaryotic and eukaryotic in all the remaining kingdoms. Cell wall is non-cellulosic and is made up of polysaccharide plus amino acid in Monera and in Protista present in some, in fungi present with chitin, in Plantae it is made up of cellulose and in Animalia cell wall is absent. Nuclear membrane is absent in Monera and present in remaining all the kingdoms. Body organization is cellular in Monera and Protista and multicellular or loose tissue in fungi, tissue and organ in Plantae and tissue, organ or organ system in Animalia. Mode of nutrition is autotrophic in Monera and Protista and heterotrophic in fungi and autotrophic in Plantae and heterotrophic in Animalia. R.H. Whittaker in 1969 proposed a five-kingdom classification. The kingdoms defined by him were named Monera, Protista, Fungi, Plantae, and Animalia. The main criteria for classification used by him include cell structure, body organization, mode of nutrition, reproduction, and phylogenetic relationship. Table 2.1 gives a comparative account of different characteristics of five kingdoms. The three-domain system has also been proposed that divides the kingdom Monera into two domains, leaving the remaining eukaryotic kingdoms in the third domain and thereby a sixth kingdom classification. You will learn about this system in detail at higher classes. Let us look at this five kingdom classification to understand the issue and consideration that influence the classification system. Earlier classification system included bacteria, blue-green algae, fungi, mosses, ferns, gymnosperm, and the angiosperm under plants. The character that unified this whole kingdom was that all the organisms included had a cell wall in their cells. This placed together groups which widely differed in other characteristics. It brought together the prokaryotic bacteria and the blue-green algae, cyanobacteria, with other groups which were eukaryotic. It also grouped together the unicellular organism and the multicellular ones. Say, for example, Chlamydomonas and Spirogyra were placed together under algae. The classification did not differentiate between the heterotrophic group fungi and the autotrophic green plants. Though they also showed a characteristic difference in their walls composition. The fungi had chitin in their walls, while the green plant had a cellulosic cell wall. When such characteristics were considered in fungi, the fungi were placed in a separate kingdom, kingdom fungi. All prokaryotic organisms were grouped together under kingdom Monera. and the unicellular eukaryotic organism were placed in kingdom Protista. Kingdom Protista has brought together Chlamydomonas chlorella, earlier placed in algae with plants and both having cell wall, with Paramecium and amoeba, which were earlier placed in the animal kingdom which lacked cell wall. It has put together organisms which in earlier classification were placed in different kingdoms. This happened because the criteria for classification changed. This kind of change will take place in future too, depending on the improvement in our understanding of characteristics and evolutionary relationship. Over time, an attempt has been made to evolve a classification system which reflects not only the morphological, physiological, and reproductive similarities but is also phylogenetic, is based on evolutionary relationship. In this chapter, we will study characteristics of kingdom Monera, Protista, and fungi of the Whittaker system of classification. The kingdoms Plantae and Animalia, commonly referred to as plant and animal kingdoms respectively, will be dealt separately in chapter 3 and 4. Thank you.